fine. I like it.
And of course, the one of the twins. Oh my god, it was just like no, and then nobody said her. That's exactly what I was expecting, that reaction. But you didn't do it. Well, it was very different. It was, I mean, out of the row right next to the half watch, it was very different. Some of them were the sons and daughters of enemies, but they were friends with, but that was weird. I thought they'd invite Churro, like some of the Commonwealth flag countries, but they didn't. But they have more runs too.
They're friends with their grandma.
Yeah, of course. It's not fun.
Ready? I'm ready. Wait for you.
We'll wait for you. No pressure, Rich.
Okay, we will get started because it's a couple of minutes past 7. So before we get started, I would like to begin by acknowledging that the land on which we gather is the traditional territories of the Coast Salish peoples, including the Musqueam, Squamish and Tsleil-Waututh nations. And secondly, to welcome you to the Vancouver Aquarium and Oceanwise initiative. Thank you for coming. For those of you who are not quite sure this video works. For those of you that have not been here before, I'm going to play you a small video so you can learn a little bit more about Oceanwise, what we do here and why we think it's so important. Or not, as the case may be. I won't, because the video's not working. But I think lots of you have already been here, so I think that's fine. So I will then remind you that Public Programs has got a new website. So to find out about upcoming community events, go to education.ocean.org/Ocean Matters, and there you'll find the upcoming lectures and so on. And the next. So, and to tell you about the next event, if you're interested, on Monday the 18th of May, so this is exciting because it's a Monday instead of a Tuesday, we will be hosting Todd McClure. He is an author and he's written 4 books now, so his newest book is The Return of the Sea Otter. And this is all about the sea otters along... Yeah, I did write that down. That's weird. Yeah, it's over. June! There you go. Good one. Sorry, thank you for that. June the 18th. So, June the 18th will be Todd McClure. He's going to be talking about his new book, Return of the Sea Otter. And he will be here, and he will be also bringing some books along that you can that he will sign. And his book is all about his journeys along the Pacific coast from California to Alaska, which he tracked the health status, habitat and viability of the sea otters. And he will share stories of his adventures, as well as outline the natural history of these adorable animals, especially if you've seen them outside. They're absolutely incredibly cute. But also they're very important because they play a vital role in maintaining the health of the kelp forests. So come along to meet him and hear all about his incredible stories on the 18th of June, despite what it says up there. And also, just before we get started, I just want to also remind you that these events are free, but they're not free to put on. They do cost a little bit of money, especially to make them as accessible as possible for everybody who wants to come and learn more about our oceans and ocean health. So if you do have a spare $1 or $2, there'll be a donation box just outside. If you could give us anything that you have clicking around in your pockets, that'd be absolutely fantastic. So with that, I'm going to introduce our speaker today. So our speaker is Sigal Balshine. She is a professor at the Department of Physiology, Neuroscience and Behaviour at McMaster University. She got her BSc in the University of Toronto, followed by a PhD from Cambridge University in the UK. She currently studies the behaviour ecology of aquatic animals. And also, I know her because she's passionate, like me, about science, outreach and science education. So she will tell you a little bit about the amazing creatures that she studies. And yeah, I hope you all welcome her.
Can everybody hear me? Nice and loud. Good evening, everybody. Turned off. Now it turned on. Okay, how strange. So I want to start off by thanking Ruth, who kindly invited me to give this talk. Ruth and I met each other about 3 years ago when I was visiting UBC, and we shared this interest in really creating science for the public and bringing science to the public. And I was delighted when she asked me to come and speak tonight. So, as Ruth said, I'm a professor at McMaster University where I run ABLE, the Aquatic Behavioral Ecology Laboratory. But our story tonight actually goes a long way back. I'm going to take you back to the 1980s, the early 1980s. And there was a community of houseboat owners north of San Francisco Bay. And they were having problems. Every night when they were trying to get to sleep, they would be awakened by a horrible sound that just kept them up. And it sounded something like this. And they would complain, they'd call the city. By the time the city workers came in the morning, there was no sound. And the houseboat owners were getting more and more frustrated and more irritated because they weren't getting any sleep. Everyone in the community had a different idea about what was making this sound. A number of people thought it was someone using a very loud electric razor. Others said, no, sounds like swarming bees under our houseboats. That's what's creating this sound. Most of the residents agreed that it sounded like a motor, and there was a couple of people in the community who believed in conspiracy theories, and they were convinced that the US Navy had secret drills in the bay and they weren't allowed to tell them because it was government secrets. But it took a biologist to figure this all out, and it turned out that the sound was created by this 25, 30 centimeter long fish known as the plain fin midshipman. So the first fun fact of the evening is that midshipmen sing or hum. They make this sound. But you might be asking, why do they make this crazy sound, and how can a fish make such a sound? Well, it turns out that it's only the male fish that make this sound, and they hum or sing to attract females. Females find this sound irresistible and love it. They use it as a beacon to find the male of their choice. Okay, that's great, but how do the males make this crazy sound? Fish have a balloon-like structure inside of them called a swim bladder. And midshipmen have a swim bladder that's modified. So most swim bladders are for gas exchange or for buoyancy. You fill it up when you want to go up, you press it down when you want to go down. But the midshipmen have this really cool modified muscle along the sides of this white balloon. And this is called the sonic muscle. And Andy Bass, who's a friend and a colleague from Cornell University, figured out how this swim bladder works Basically, it's like a tight drum full of air, and the muscles vibrate really, really quickly, and they make this humming sound. And Andy went further. He even figured out where in the brain this muscle is controlled. And it turns out that there are these specialized neurons in the brain. They're called motor neurons, and they actually innervate something called the sonic nerve, which moves down into the sonic muscle. And Andy's mapped out the whole vocal network of the brain. And he's figured out all kinds of really cool things about these fish and how they vocalize. So they don't just hum, they actually have a whole language. Some of them will grunt. So if I go down to the beach and I listen, I can hear sounds like this. Oops. Sorry, that's a grunt. It's C. So they can make these grunting sounds, and they can also growl. And my growl isn't working, so I'm going to pretend to be a midshipman and tell you what a growl sounds like. But it sort of sounds like this. And these sounds can go on for minutes and even hours. What's incredibly interesting, I think, about these fish vocalizations is that these fish, like other vocalizing animals, will use sound to court, to attract the opposite sex, but they also use sound to repel competitors, to say, this is my territory, stay away. And we can listen to the way this fish makes its sounds. Let's listen to that grunting sound again, which is a aggressive vocalization. Let's listen to a frog make its territorial and courtship calls. And now let's listen to a bird. Quite different. And here is a squirrel monkey. Each of these vocalizing animals uses a very different organ to make these sounds. The fish, as I told you, uses a swim bladder. The frog and the mammal use a larynx, and the bird uses something called a syrinx. But what's so interesting is that the control regions in the brain that control these sound-making organs actually originate in the same place in the brain. And if you look at larval animals, birds, mammals, fish, frogs, these neurons all start off in the very same place at the back of the brain, in the hindbrain. And so this tells us that this tree of life that we're looking at here shows conservation across all vocalizing tetrapods. So the plain midshipman is a singing fish, a humming fish. But in the next few minutes, I hope to really impart on you that this fish is cool for a whole other reason as well, many other reasons. So the next thing I want to tell you about is how this wonderful creature got its name. Lots of people ask, why is it called the plain fin midshipman? How did it get its name? And it gets its name from these light-emitting cells on its ventral, its belly, and the ventral surfaces are just covered with these light-emitting cells. There's over 700 of them on the tummy. And when people first saw this species, they immediately thought of the buttons on a midshipman uniform. A midshipman is a rank in the Navy, and those brass buttons made people think of those photophores, or those photophores made people think of brass buttons. Now, these photophores are in themselves a mystery to scientists, because why would a fish need 700 light-emitting cells on their tummy when most of their lives they look like this? They're deep in the mud, in the substrate. So what are these photophores for? Why would a fish that spends its life deep in the in the substrate, where all you can see is a pair of eyes and maybe a big fat mouth, why would they need ventral photophores? What is that all about? And we really don't know. There are a lot of different ideas, and scientists are still arguing about these ideas. One of the ideas is that the fish actually leave the sea floor bed at night. They'll come off the sea floor bed, and they'll use these photophores to attract prey. Just like a moth will come to a flashlight, there are lots of little microorganisms that will come to these light-emitting cells, and then these ambush predators, the midshipmen, can grab them and eat them for dinner. There are other ideas too. The photophores also, as you can see, really break up the silhouette of a fish. It no longer looks like a fish, it looks like parts of a fish. And any predator underneath the midshipman while it's feeding off the sea floor that looks up no longer sees a fish. So maybe it's a predator avoidance strategy. It breaks up the pattern of a fish. It's called counter-illumination, and it's an anti-predator adaptation. So we don't know why they have photophores, they just do. They're really, really common. They're found all over the Pacific side of North America, from Alaska down to Baja California. And amazingly, for about 8 months of the year, these fish live in the deepest, darkest part of our ocean. They'll move from 400, 300, 200 meters down, all the way up. They make this massive vertical migration up to the shallowest part of our ocean, the intertidal zone. The males do this first, and then a few weeks later, the girls follow. And they'll come to beaches much like this one, which are full of rocks, and they come from very, very deep down. And when they get there, they duke it out. The males will fight one another for access to one of these rocks. And the wonderful thing about midshipmen is that they come to us. So I can put on my boots, my rubber boots, and walk along the beach, turn over rocks, and study these fish. I don't have to put on a snorkel. I don't have to put on my scuba gear. I can study them right on the beaches, which is very convenient. And they're very, very dense. So in a beach like this, there can be 300, 500, 600 fish, which is incredible. So I told you that they compete. How do they compete? Well, they bite each other, they ram each other, they do these things called mouth fights where they wrestle with their mouths. And I have a really bad video which I took just to show you what a mouth fight looks like. It's just a few seconds, but you'll find it, I think, it'll probably make you a bit seasick, but we'll do it anyway. Sorry about this. Can you see some wrestling fish? They're right there. Yeah, I'm not a great videographer, that's for sure. Males fight for a nest, and when they get a nest, they're not just satisfied with taking that nest, they'll modify the nest. They spend a lot of time digging it out and making it home sweet home. Here's a little fish who's preparing his nest. Whoops. Let's see if I can do this. And that video was taken by one of my graduate students who's much better at videography than I am. So I told you that the males use these incredible hums to attract females. But it turns out that not all males sound the same. Bigger males can sing louder, they can sing lower, and they can sing for longer. And the girls really like the long, loud, lower sounds. Here you can see some tiles. We put out tiles in the intertidal when we're doing experiments because we want to standardize in our experiments nest sizes. And this male received a lot of eggs. The girls really liked him, and many girl fish came and spawned with this male. And on the very same day, on May 12th, I think, this poor male got very few girls. Maybe one girl came and spawned with him. The girls have about 100 to 200 eggs, and you can see that this male attracted many females while the poor guy on that tile got very few. And this is probably because his sound wasn't as nice. Now, once the girls lay their eggs for these midshipmen, they leave. They go to sea, never to be seen until the following year. And the poor guys are left taking care of the babies all by themselves. And they stay there for about 4 months looking after young. They'll defend them from predators. They'll clean the eggs, blowing water and fanning the eggs, making sure they have oxygen and they're clear of sediment. And they'll perform hygienic duties, removing any egg that's diseased or has fungus on it. And these babies take an awful long time to develop. 60 days in all. 30 days until they hatch, and then another 30 days while they're absorbing the yolk in the egg, and they're finally ready to go off after 60 days. So right over here, wait, maybe I can do this with my pointer. You can see a baby fish, but he still has an egg, and he might be on this nest for another 2 weeks, absorbing the delicious nutrients in that egg before he's really ready to go off to sea. And if we open up nests, what we see is different development stages of eggs. These are newly hatched eggs, and they get more and more orange as they develop. Eventually they hatch, and you see little baby fish, and after 60 days, they're all silver and ready to go out to sea. They're baby midshipmen. But the males sometimes, after the 60 days of care, are lucky enough to get a brand new clutch, and so they can stay there for yet another 60 days. Here you can see such a clutch. This male here has a nest full of young that are about to pop off the eggs, off the nest, and go off to sea. But he was lucky enough to attract another female or two, and now he's stuck here for another 60 days. And they can't eat. There's nothing for them to eat. So they're starving for 4 months. And when we first start the season, our males are very bulky. They're fat. And as the season progresses, they get skinnier and skinnier. Looking after baby midshipmen takes a lot of time and a lot of energy. And my research group has been engaged in doing all kinds of experiments to measure these costs of care, these extreme costs of care. So we can measure fish on the beach using scales, and we can see that as the breeding season progresses from May to July, these fish lose body condition. They lose weight, they're in bad shape. We can also test their tissues and their blood, and we can see that the fuels that energize a fish, things like glycogen and glucose and proteins and ATP, those also reduce. And when we experimentally starve some fish in experimental tanks, they're very similar to those fish we find on the beach in July. They have very few fuel stores left. Now, I told you that they come to the intertidal zone. So this makes caring even more costly, because not only are they there for a very long time, but they're caring under really crazy conditions. They're under these rocks even when the tide recedes. They remain under these rocks. Here's a fish nest, here's a fish nest. There are a bunch of different fish nests marked with a tent peg, and the males will be under these rocks, still caring even though the tide has taken the water far away. They'll stay under these nests, sometimes air exposed, for several hours, up to 8 hours. Some fish are lucky enough to have a little tiny pool of water under their rock, but others dry out completely, and they tolerate this month after month. So males are caring in an extremely challenging and very dynamic environment where environmental conditions are fluctuating. The amount of oxygen is fluctuating, temperatures are changing, salinity is changing, you name it, these fish are having to deal with this. And you thought life was tough, but just you wait. They also have to contend with predators. So the birds love midshipmen, and they eat them constantly. So these guarding males also have to contend with things like herons, crows, eagles, and gulls, sometimes who are feeding exclusively only on these fish on these breeding beaches. And each of these bird species actually has their own special way of dealing with the midshipmen and how to process them and handle them. Midshipmen have spines on their opercula, on their gill covers, and these birds don't want to get stabbed by these spines. So they each have their own way of dealing with it. Suffice to say that midshipmen are a really important part of our marine eco food web. So if you look, midshipmen eat squid and krill deep in the ocean when they're there. But then when they come to shore or on their way there, they can be eaten by things like porpoises, by all kinds of shorebirds and water birds, things like sharks and seals all eat midshipmen, and large numbers. So this is a dangerous caring situation. Dads are caring alone, and it's a very costly kind of care. Now, the story gets even more interesting, because some dads manage to avoid care altogether. So there are actually two kinds of males in this picture. There's a big caring male in this photo, and down here you have another kind of male, and this male is called a sneaker male. There are these two types of males. In this picture we can see the big caring male, some babies, a female, and a tiny little sneaker male. They're sneakers because they're sneaky. They're little and they try not to be noticed. Look at how different they are. Big, large garter males, tiny little sneaker males. We called this guy Rambo just for a joke in the lab. So how are they different? Well, the garter males, they're very large and they're very vocal. They make these wonderful hums. They grunt and they growl. They compete, they bite, they mouth wrestle, and they build nests. They court females and they care for young. They're kind of your hero dads. But we also have this second kind of father in the population. They're not such good dads. They're small, they're silent, they never compete, they run away from competition, they never provide any care. All they do is they sneak in, drop their sperm, and dash out. So these guys get away with murder. They basically are very, very different in every way from the garter males. And our lab has been wondering for a while why there are these two kinds of males. Why should the species have evolved these two types of males? So one of my previous graduate students, Karen Cogliotti, who's now a faculty associate at the University of Oregon State, she decided to address this question. And she wanted to know, well, how common are these sneaker males? And is sneaking really a very successful strategy? Maybe it's not. So Karen decided to address this question by looking at how common the sneakers are over time in the breeding season. Are they common all through the breeding season or only at one time? And also across the range of these midshipmen, across space. How did she do this? Well, we have many, many different study sites, many, many different beaches where we can study these fishes. And so what Karen did is she lay these long transect lines, 100 meters of line down on our different study sites. And then every 10 meters, she put out what we call a quadrant, which is basically just a big square. And she mapped out all the nests in these quadrants, and she turned these quadrants into her kind of study of how common sneakers are. So every time Karen found a nest, she would mark it. She would mark it with a tent peg so we could come back to it, time in and time out. She also measured all the fishes on the beach. She could put them on scales, she weighed them and measured their length. And she also cut, very importantly, a little tiny bit of their fin tissue so she could get a DNA sample from these males. She also injected the males with a kind of tattoo dye that's called elastomere. And this leaves a permanent mark on the male so we can track them over time. And finally, very importantly, she also took a sample of eggs from each nest so she could match the fin tissue from the dad to the DNA in the young. And so here's a great picture of one of our males with his tattoo markings. And you can imagine that every male can have their own ID. Sorry, is there something wrong? No. This is the ID, exactly. And what Karen found was that sneakers were far more common early in the season compared to the middle and late in the season. By July, there were hardly any sneakers around at all. And Karen also studied this question of how common the sneakers are in our different study sites. She looked at 5 northern sites in British Columbia and Washington State, and then 4 sites in the south in California, using that transect and quadrant method to figure out how many sneakers there were. And the results were quite surprising. We found that sneakers were rare. There was about 1 sneaker for every 9 guards, and that was true across our populations. So in the south, about 91% of the fish were garter males, of the male fish were garter males, and in the north, the same thing was true. So sneakers are rare. Now, she also, as you recall, took some eggs and some fin tissue for DNA work. And what scientists do when they want to figure out paternity is they often use something called microsatellites, which are special molecular markers that were developed for this fish to figure out who's the dad of which young. And it very much is just, there's basically peaks in that the analysis makes from the genetic material, and you look to see shared bands. So here's a mom, and she shares a band with this child, so the child got this band from its mother. But it got this peak, number 7, from the dad. And using these kinds of techniques, scientists can match up the young to the putative father who is looking after these young. And when Karen did this, we learned something that really shocked us. Only about 52% of the eggs in a male's nest were his own. 48% of the young he was looking after and providing all that expensive care were for young that weren't related to him. So sneakers are a lot more rare, but they are more common early in the season. They're generally not common compared to garter males. And garters are only related to about half of the young in their nest. So, given that care is so costly, we were wondering, are males really going to be willing to care for young that aren't their own? If you have an obvious signal that these are not your puppies, should you be defending them? Should you be caring for them? How should a male respond to low paternity? One of my other graduate students, Anish Bose, who's now a postdoctoral fellow at the Max Planck Institute in Germany in Konstanz, wanted to ask this question. He wanted to ask the question, do garter males really respond to low paternity? Maybe they're there, but they're not giving really good care when the kids aren't theirs. We had a whole bunch of predictions, or better still, Anish did. He thought that a male might reduce the amount of parental care it provides to young when the young are clearly not his own, and they may do that just by not fanning very much, not defending very much, just chilling out under the nest, just not working as hard. So we thought maybe they just simply reduce care. More extremely, maybe they eat the eggs. Maybe this is a competitor's eggs, maybe they start eating them. Or maybe they'll just abandon altogether when the paternity is low. Really what we were asking is, what cues, what signs could a parent possibly use to detect that they've been cuckolded, that these eggs are not their own? that they've lost some paternity. The fish can't go out and buy a DNA kit like you can for $119, but the fish might need to respond to these kinds of insults as well. So what Anish did is he ran a bunch of experiments in the lab and the field to try to figure out how paternity and changes in paternity affect the fish and their behavior. And to do this, he used these artificial nests I told you about. Basically, they're just tiles. He threw concrete tiles out into the intertidal zone. And the fish love these tiles. Within about a day of putting out tiles, we have males burrowing down, females spawning under these tiles. They absolutely love the tiles. So Anish's first question was, how might a male assess that he has high paternity, low paternity, that these eggs are his? And he wondered if they were using some kind of cue, some kind of signal from the eggs themselves. Animals can sometimes hear their own babies, smell their own babies, taste their own babies, or visually recognize their own babies. And we were wondering if these fish might be capable of something like that. Maybe there's some direct cue emanating a smell from the offspring that tells the dad, these are my babies. So what Anish did is he ran this experiment where the tiles were either part of a control, where he simply lifted a tile and switched it around 180 degrees just to control for disturbance. And he had another experiment where he transplanted tiles. So a dad would get a complete foreign brood in his nest, none of his own young. And then we watched how these fish survived, depending on whether they were looking after their own broods or somebody else's broods. And sadly for both of us, there was really no difference. So this is survival, the proportion surviving after 3 days, after 14 days, after 28 days, there's only about 40% survival. But the black line, which is the fish that had their own broods, and the fish that had foreign young were almost identical. There was really no difference. So we were wondering, do they even know that there are eggs there? Are they even noticing that these are different? What's going on? And so to test this further, Anish built these beautiful aquarium experiments where he gave fish a choice between 2 tiles. And this was all done at the Pacific biological station in Nanaimo. In the first test, the fish were either given no eggs on a tile or eggs on a tile. And these were always foreign eggs. And we asked, did they go to this tile or did they go to this tile? And then another batch of fish were tested between their own brood versus a foreign brood. And it was the second test that was the critical one for our understanding of whether or not males can detect offspring cues and know that they've been cuckolded. So firstly, what we found is that males almost always went to the nests that had eggs. They preferred to go to the tiles that had eggs over here versus the control or empty nests. So that's good. The fish obviously could detect eggs. But in our critical test, when we asked, did the fish prefer their own broods versus those with foreign broods, the answer was not really. It was just chance. They equally divided themselves up between their own broods and those of someone else. So to summarize, what we know so far is that males definitely prefer nests with eggs, but not necessarily their own eggs. And males don't appear to be responding to any direct paternity cues from the offspring themselves. Now, maybe there are other cues. There's another male in my nest. There's a sneaker here. I smelt a sneaker here. There could be other indirect cues, situational cues, that determine how these fish behave towards their eggs, and we're exploring that right now. But just as a kind of take-home message from this talk, and things I want you to go away with and remember, are that midshipmen are really interesting because they sing. They hum, they make these incredible sounds, they grunt, they growl, they have a language. They also use light either to attract prey or deter predators, but they can do this. It's really incredible. And they can tolerate extremely long periods out of water, 4 hours, 5 hours, 8 hours out of water for long periods. They care for their babies for 4 months and they starve during this period. There are 2 kinds of males. There are good dads and there are cads, the sneaker males. These sneaker males are far less common, and males are definitely losing a lot of paternity to other males. Males don't seem to be responding to any of this paternity loss and will continue to look after foreign young. So if that didn't convince you that this is a very interesting species, I want to leave you with a few more thoughts. I want you to think about these midshipmen and how lucky we are that we have them, and why they're so interesting to me comes from some of these reasons. These are really fish that provide us insight into ocean health. They're a kind of canary in the coal mine. They're a sentinel for ocean health. These are fish that spend most of the year feeding in the deepest parts of the ocean, which are really hard to access and study. And they make their own migration to us in the intertidal zone. We don't have to work hard. We don't have to chart a submarine or some kind of device to go into the ocean and figure out ocean health conditions. We can measure their blood, look at their livers, look at their muscle tissue, and see what they've been eating in our oceans, and what the health of these fish is going to tell us something about the ocean's health. They also are a modifier of the intertidal. So they're not just satisfied taking any old nest, they change it. They pull off all kinds of creatures. They create space for their eggs. But in doing that, they create space for many other creatures' young. So lots of small mussels and oysters and clams and our acorn barnacles will take hold after a midshipman has left a clean, beautiful nest open in the very crowded intertidal zone. Finally, there are lots and lots of commercially important fish. Midshipmen are not commercially important. You wouldn't eat one. They look kind of ugly. They look like frogs. But there are lots of fish that you probably care about more because you like to eat them or fish them or whatnot. Things like salmon that have identical reproductive tactics. Salmon have, instead of sneakers, we call them jacks. Instead of garters, we call them hook noses. But they're basically the same alternative reproductive tactics. The ways of getting reproduction vary between males. And to date, people really haven't considered how these alternative reproductive tactics influence population health. But our lab likes to think that we should be doing that. So for these 3 reasons, the fact that they're a sentinel for ocean health and are part of the food web, because they're a modifier of the intertidal and create opportunities for more biodiversity, and because there's so many important commercial fish that have similar alternative tactics, reproduction tactics, we need to understand more about their dynamics. So with that, I'm going to just end with a bunch of thank yous. Thank you to you for listening today, and thank you to the funding agencies and to the many, many people who made this study possible in many, many different ways. And with that, I'd love to address any questions that you might have.
So thanks, Sigal, for that incredible talk. And I have a mic here for people who want to ask questions.
I have 2 questions, but I'll start with my first one and see how the second one goes. So these sneaker fish, are they fish that survive and they go out and they grow and they're just small enough or big enough to come back and be like, hey, I know I can take advantage of this? Is that kind of how it works?
So you're asking the million-dollar question. Do sneaker males grow up to be garter males? And the answer is we don't know. We don't know. We'd love to know. We mark the fish and we never see them again. So we mark them and they're fine on the breeding sites for about the breeding season, and then once it's over, we never see anybody again. Sneaker, female, garter male, we never see them again. I actually have a sneaker and a garter, and I'll pass it around. This is a garter. And this is a sneaker, but he's a big sneaker. He's quite a big one. So I'd love to answer that question. Maybe you can think of something that we could do to address that question. We would love to do that. We're trying to grow them up in the lab and see if sneaker dads have sneaker sons and garter dads have garter sons, because that would be one way at least to partly answer the question.
Well, I would like to talk to you after about some research endeavors. Second question is, you said that the fish, the midshipman fish, are sentinels of the ocean. Have you found in researching or investigating their cellular content, blood content, liver content, a spike or an increase in the amount of plastic or microfibers, microplastics?
So we have done a lot of diet studies, but at the time I didn't have my microplastics lens on. So I have those gut contents and I can go back and check. I can tell you that their tissues show that they are contaminated. They're not necessarily contaminated with microplastics as far as my naked eye can tell, and the microscope work didn't really reveal a lot of plastics, but I didn't have that lens on. But they have furans, they have dioxins, they have PCBs, they have a lot of nasties in their tissues. And so that suggests to us that something's not quite right, and we'd like to know where they're picking up those contaminants. We're not sure.
Yeah.
Shh.
Thank you. So, what you were saying, that you never see the fish again, do you suspect that they only reproduce once? The way
So I don't know who I'm talking to.
right here.
Oh, okay. Thank you. There is suggestions in the literature that that is what happens, that they have sort of big bang reproduction, much like our Pacific salmon, and they never come back. There are two reasons why I don't necessarily believe that that's the case. One is that if you look inside the female's ovaries, she'll have the eggs that she's laying that year, and she also has microscopic tiny eggs that won't mature for another year. So why would she even have those if she's not going to come back? So at least the girls come back to live another, breeding day. The guys, I don't know, but we have males that are quite small, that are garters, and then we have big honking guys. And those fish are different ages. So I don't know. I don't know. I would love to know. I think they're really tapped out by the end of the breeding season. And I wouldn't be surprised if some of them don't make it to come back, that they're just too worn down.
Right. So
Many die, but I don't know if all die.
Because I think Mark Gross found that the jacks actually are faster growing than the hooknoses. Do you suspect the same thing, that these are actually, that the sneaker males are actually the faster growing phenotype?
So the way to tell how fast a fish grows is to do usually an otolith experiment, to look at their ear bones, because they have rings like trees that tell you how fast they grow. We've sent these fish's otoliths to 4 different labs around the world, and nobody wants to tell us how old they are. They say they could be 27 years old or they could be 3 years old. They have very weird, long otoliths that are kind of really notched and look strange. And nobody's willing to give us an age. But when they do compare the sneakers to the garters, they say that the sneakers are younger. But no one's telling us anything yet about growth. So I don't know, but I'm really glad you mentioned Mark Gross, because he was my inspiration. He's the reason I became a behavioral ecologist and a fish biologist. He inspired me.
Oh, it was the same. Anyone else?
I just want to ask a couple of questions. The first one being if they stay around the tide pools and on land and effectively dry out for possibly hours.
Yes.
do they have like something like a primitive lung, or is there any other way they survive that?
So some fish have evolved air breathing, and we suspect that these guys can air breathe a little bit. We're doing studies right now to look at that. We also think that they use their swim bladder like a scuba tank, an internal scuba tank. Why do we think that? Because early in the tidal cycle, when the tide's just going out, if we measure the oxygen in the swim bladder, there's a lot of it. And then as they spend longer and longer out of water, the amount of oxygen or air in their swim bladder is reduced. And so we're studying this right now in greater detail, but they do have a lot of adaptations to deal with this hypoxic air exposure environment. For example, they are hunkered down and they almost go into like hibernation mode. They don't move at all during this period. They're very, very still, not wasting any energy.
Has there been an average or time?
The average time out of water?
Yes.
So that depends on where the male is in the intertidal. Some of these males will make their nest subtidal, and then they're always in water. Others will come out just a little bit, and they're in the low intertidal, and then they may be out of water for 2 hours. But there's other guys who are way up high in the intertidal, experiencing 7 or 8 hours of air exposure, dryness, no tide. Why? Why do they do that? This is another question that we're actively pursuing, and my hypothesis right now is that their babies grow faster in that warm area under rocks that are air exposed for a long time. But I can't publish that because it's not true yet, but I hope to show that it is.
Thank you very much.
Yes, I've got one question here. When you were referring to the, just at the ears in terms of
The otoliths?
the otoliths, and them predicting how old a fish is, have you been able to determine the length of lifespan of those fish?
So sadly, this is where labs are refusing to give us a clear-cut answer. They were saying that the rings are not normal-looking rings for a fish, and they are uncertain if they're short-lived or long-lived. So we don't know.
Thank you. Yeah, great talk. Thank you. Just curious, wondering, I'm fairly familiar with the story of the midshipman making sounds on San Francisco in the 80s. Can you say anything about what's going on with them now in terms of how acoustically active they are and how they're impacted by underwater noise pollution?
Yes.
If that's a significant...
So we're currently doing studies with a colleague called Francis Wanis, who's an expert in fish sounds and acoustics at the University of Victoria, and we've been putting hydrophones out in many of these fish's breeding areas. And what we can tell you so far, we've submitted a paper, it hasn't been accepted, but it's showing that in areas where there's marinas and a lot of boat noise, the fish actually change the frequency of their call. Not a lot, but a little bit higher, yeah. And so it's interesting. Now remember that these fish are nocturnal. So they're calling at a time when there isn't as much boat traffic. If they were in the day, calling in the day, I'd be more worried about them. But because they're nocturnal, usually the boat noise is at a minimum at the time that they're calling, like 12 midnight to 6 a.m. So that might actually help them.
Okay, and when you say the frequency is changing, can you actually specify what frequencies?
I could if I looked in my computer, but I can't off the top of my head. I'm just holding a lot of different facts, and I'm not an acoustics expert, but Francis is, and I have it. I could answer you if I looked it up. Or I can send you the paper.
Okay, so can you say anything about how the populations are doing overall for these guys?
So They're doing great in most areas. They're extremely common on rocky beaches. They're easy to find. But there's one area where there's a bit of a decline, and we're worried it's due to a pulp and paper mill. Around Ladysmith Inlet, there's a population that seems to be in decline, but overall, most of our populations are stable.
Okay, great. Thank you.
Any more questions?
At one point we discussed in a biology class that there was a hypothesis a long time ago about the lemmings, and the lemmings who would, sacrifice themselves for the good of their species.
Yeah.
Has there been any studies done with these fish in regards to the fact that they're willing to take care of another man's kids, regardless of maybe for the good of their species?
So that came out in a Gary Larson cartoon. I don't know if you've ever seen that Gary Larson cartoon, but it shows a bunch of lemmings jumping off a cliff, and one of them's wearing kind of like a safety belt so it won't drown. And it turned out that that story was false. Lemmings can swim, and so they don't jump off cliffs for the good of their species. Animals don't do anything for the good of their species. They do things for the good of themselves. So we usually think about animals doing things to maximize their own reproductive success, and that may just happen to improve what happens to the population. We're not exactly sure why these males continue to care for other males young. But there could be some advantages, like maybe you dilute the predation risk for your own young by having other young around. Or it may be that females are actually attracted to nests that have a lot of eggs, and so by having extra eggs around, you may actually get more girls. We're actively looking into this. It's a mystery. We don't know yet, but that's what science is exciting, what makes science so exciting. We don't know the answers. Every time you get an answer, it opens up 5 other questions for you to answer, and this is one of them that's definitely opened up as we've figured out that they care for other males young. We thought they wouldn't. We thought they'd like just abandon or eat them or stop caring.
Are there lots of people worldwide studying these?
No, there's about 5 labs in the world that study midshipmen.
Is it mainly North and South America, along the range, along the west coast?
So this toadfish doesn't go further down, like it stops at Baja California. So most of the labs that are studying this fish, well, let's see. There's my lab, Francis's lab at UVic. There's a lab at the University of Washington in Washington State. There's a lab in New York City and another lab at Cornell. Those are the 5 labs that study this fish intensively.
And all these 5 labs, has anybody raised them successfully for any period of time to get an idea of...
Up to a year.
Okay.
We've only managed to raise them for up to a year.
And then they pass away, or have you just...
So we haven't even got them to get to 5 months. They don't seem to thrive under our care. They need something in the ocean that we are not providing. But Andy's lab has managed to keep them going for a year down in California in one of the labs, the aquatic labs there. We're not sure why. We've been picking his brains. He can't figure out why they lived. And it was in the early 90s, and no one's been able to repeat it since.
Is there any way you can set up a net pen type idea? Very fine mesh or some type of box system?
Yeah, that's a great idea. I think they're very much connected to eelgrass ecosystems. They release their babies in areas close to eelgrass, and I think eelgrass is key to their survival. I'm not sure if it's food there or hiding from predators and decreasing stress, because the eelgrass provides them from protection from predation, but I think it has something to do with eelgrass. So we're trying to mimic eelgrass now in the lab and see if we can get them to grow up.
Sorry. The other thing I wanted to ask you about, you had said that this particular fish goes from the north of Alaska all the way down to the Baja of California. And I know that particular route, there's lots of ponds of pools. And this fish, does it typically, when it comes into the tidal portion, is it where those tidal pools are?
So tidal pools can be in exposed areas and in protected areas. And they're always found in protected areas. So they can't have massive waves or surge because they need to make the sound to attract the girls. And if the waves are louder than your call, that's no good. So you find them in really quiet places that are really devoid of big, huge waves. They're not found in Oregon for some mysterious reason. I've looked for them in Oregon and never found them.
I think we have time for just maybe one or two more questions.
Could you comment on the anatomy of the mouth? Fascinating.
They have big mouths. Yeah. So I love their teeth. They are so fun to watch through the breeding season because they use these teeth to fight with one another. And you'll find males with big, huge gashes and holes and ripped fins. And by the end of the breeding season, the males look like old men without any teeth. Their teeth are all worn down. Yeah, those are actually the gill arches. So they're holding up the gills, which you can see behind. It's a bit of red.
So I think just one more question.
But they might use those gill arches to capture food as well.
I'd like to ask about when you said you couldn't keep them alive in the labs. How would they die? Would they waste away, or like wouldn't eat, or would it be stress factors?
I hate saying this in public, but yeah, they were starving. We tried everything. We fed them everything we could think of, and they refused to eat it. It was very sad. I really hated that, and I hate admitting that in public. I'm probably on TV now. Oh my God. Can you cut that part out of YouTube? Oh God.
Magic of editing.
But just for the record, my students as well try to raise spiders, and it's really difficult if you the complex elements can be tough. Maybe we should have one more cheerful question at the end.
Cheerful question, no pressure.
I was just wondering about the different eggs that were in one male's
clutch or brood.
Nest.
Yeah.
Were they? I don't know if you know, but were they just from the sneaker males, or were they from other big males? And like, do you think just the eggs from sneaker males aren't detectable for them, or I don't know?
That's a great question. So we know that sneaker males are actually not getting that many of the fertilizations. About anywhere as low as 3% to 17% of the eggs go to of the young that have the male lost paternity goes to sneakers. The rest actually come from other large garter males who try to act like sneakers when they don't have their own eggs. So when a male doesn't yet have his own eggs, he'll come to a male who has a girl and has some eggs, and he'll try to sneak as well. So they just go for it.
Okay, well, thanks, Sigal, for that amazingly interesting talk, and thank you everybody for coming along, and hopefully see you at the next talk, the 18th of June. Okay, thanks.